All right. Well, welcome everybody to the second part of our speaker series. Series is put on by the student chapter of Society for Ecological Restoration here at the University of Oregon. We're a new student group, and we have our series on sourcing plant material for restoration under climate change. And we have two speakers today. I'm going to introduce the first one. Gabby will introduce our second speaker. And I think we're just going to kick it off. So our first speaker today is Dr. Sally Aitken. She's a professor and associate dean of research and innovation at the University of British Columbia. Sally conducts cutting-edge research in forest conservation genetics with implication for large-scale reforestation practices in the West. In 2001, Sally started the Center for Forest Conservation Genetics at UBC. In 2009, she was awarded the Canadian Forestry Scientific Achievement Award. In 2012, she coordinated the BC Big Tree Registry, an online registry for documenting big trees in British Columbia. In 2017, Sally was elected a Fellow of the Royal Society of Canada, and a year later, she was awarded the Genome BC Award for Scientific Excellence. Sally's going to talk to us today about her research. We'll have about a 15-minute talk, and then we will switch over to our second presenter for another 15-minute talk, and then have hopefully about 20 minutes at the end for questions. And with that, I will put myself on mute and get started.
Thanks very much, Alejandro. And it's really my pleasure to be here. I am on an island called Quadra Island, which is between Vancouver Island and the mainland. And I'm in the traditional unceded and ancestral territory of the Kaye First Nation and the Coast Salish people. And I really love that. The only thing that I love about the pandemic is I've been able to spend the whole time on this beautiful island and a lot of time out in the forest. But research goes on, and so I'm going to talk to you today about our work on trying to inform choices around what to plant in a changing climate. And so I'll be talking about some results from large-scale projects that we've done called Co-Adaptree and Adaptree, as well as some other work. And this is all on trees. It's mostly on, actually everything I'm reporting today is on conifers, although that's not all that we have worked on. But I hope some of it is of interest to those of you interested in other plants and other types of restoration activities. So I don't need to tell you that climate change is already affecting forests globally, and trees are at particularly high risk of maladaptation just because their generation lengths are so long. So the amount of climate change that really matters from an evolutionary standpoint is the rate per generation, and long-lived trees are experiencing a faster rate per generation than pretty much anything, except maybe large soil fungal clones. So in BC, a lot of work's been put into climate change projections. Just for those of you who aren't familiar with BC, it's in green on this map, and it's about the same size as California plus Oregon plus Washington. So I'm going to show you a lot of maps that are just BC, and that gives you an idea of their scale. Some of those maps will include Alberta as well, which is nearly as large as BC. This is a map of our current ecological zones in BC. There are 16 zones, 13 of those are forested, and actually 12 are forested. And at the bottom here, we have a couple of different runs of different GCM ensemble runs for the 2050s under RCP 4.5, so moderate, sort of better than we might expect climate change scenario in a lot of ways. And all I want you to look at is the extent to which the green ecosystems have expanded relative to the other colors. if you focus on any one color here, you will see a very large change in distribution. These green ecosystems happen to be our coastal Douglas fir, so coastal temperate rainforest, and our interior temperate rainforest, which is the interior cedar hemlock ecosystem. But we're expecting a massive redistribution of the climates associated with our major ecosystem types, and shifts in the climate envelopes for individual species. And so we're expecting within our BC context, some species are going to lose habitat, like lodgepole pine. Here you see projections of the changes in suitability of areas for lodgepole pine. If they are in red or pink, these are areas that the feasibility is dropping towards the 2050s. And if in blue areas, the feasibility, the model feasibility of the suitability of the species is increasing. So lodgepole pine in our context, which is our most planted species out of our 300 million trees that are planted a year, is going to be losing ground. But then we've got Douglas fir and others that are expanding. Western red cedar would be the same way, where we see areas that the species has been it's been too cold. These are modeled to warm enough to support this species, which of course is the core of the forest industry in Oregon, as well as coastal BC. So as we think about the natural response of populations to climate change, we may see migration from the leading edge into areas that are becoming suitable. We may see adaptation over generations based on standing genetic variation within populations. And we may see contraction of the range at the rear edge as the climate envelope shifts, the climatic niche shifts over the landscape. And how quickly these populations will respond is a good question, and we're working on that from genomic data. But it's very hard to predict when these rear edges, if and when rear edge populations will collapse, but we need to capture those genetic resources for restoration, for reforestation. So what can we do? Well, we can think about assisted species migration into areas that become climatically favorable. And when you say assisted migration, that's normally what people think of. But most of our work focuses on what we call assisted gene flow. You might call it climate-based seed transfer, which is all about just changing where we get seed from, changing seed sources within range. And so that might involve moving things out of areas that are becoming unfavorable. But the real goal here is to translocate individuals so that we facilitate adaptation to local conditions. And those that can be adapting to change that's already happened in climate, and it can be anticipating future climate. In British Columbia, we have moved to entirely a climate-based seed transfer system, although the policy was introduced 2 years ago and people still can use the old system if they want, I think maybe for another year. But most of the shift in what's being used for reforestation is making up for warming that has already happened. So the models target predicted climate in 15 years. So most of the shift is making up from change since about the 1930s, which is about 1 degree Celsius of change in most of our areas. So assisted gene flow then is about moving things within range. And when we look at the distribution of our widespread conifers, we see big areas of overlap where species currently are, the existing range and the future projected range. So this is some modeling done University of Alberta by Andreas Hammond's group. And this is a present day range map for lodgepole pine. And this is the projected distribution of the climatic niche in the 2050s. The yellow areas are areas projected to be already lost from the range. And you can see some expansion, but mostly these maps are reasonably similar. And the same goes for the species complex that we call interior spruce, which is white spruce, Engelmann spruce, and in British Columbia, extensively hybridized populations. So we have a long history of common garden experiments, field-based common garden experiments in forestry, and we call them Provenance Trials. And here's an example of a Provenance trial for Sitka spruce. We've just cut the trees out that represent the average of each provenance because we had to thin the experiment. So these were all planted at the same time, seedlings planted at the same time. And you can see here we have the California population that had a mean annual temperature of about 12 degrees Celsius, grew much, much faster than the colder sources, the colder provenances from Alaska. And we see a general trend here that warmer provenances grow faster, colder provenances grow slower. And the flip side of that is we see greater cold hardiness in the provenances from colder locations, which makes sense. So this is the trade-off we see. This is what drives in the forestry context, the shift when climate-based seed transfer. We want trees to be well matched to their climate so they're healthy, but also means for the forest industry that those trees will grow a little faster. And so we've had these traditional experiments, and the first Provenance trials were put in more than two that we know that there's a record of were put in more than 200 years ago. Long history in forestry of Provenance trials. And so seed would be collected from some portion of the species range, or maybe the whole species range, and planted in a series of field experiments. These maps happen to show one of the largest provenance trials in the world, which is for lodgepole pine in BC. But Darwin writes about a provenance trial at Kew Gardens that included Himalayan pines and also Himalayan rhododendrons on the origin of species. So it's been going on for a long time. But those experiments take a long time to get data from. Those field-based experiments, usually you want them to grow for 10, 15, 20 years or more. And with climate change, we've got a lot of pressure to get data faster. So we tend to use either controlled environment experiments, like was done with these spruce seedlings, where they were grown in growth chambers under different climatic conditions, ranging from a mean annual temperature simulated of about 11 degrees to a mean annual temperature simulated of about 1 degree, and different moisture and heat stress treatments. Now, across this range of treatments in the growth chambers, you can see a lot of phenotypic plasticity, in that the trees grew much faster in the hot wet than in the hot dry, than in the cold wet rather. And we can also see injury from the drought and heat stress. These growth chamber experiments are good for showing us some things, but we also find they're pretty artificial environments, and we don't always get the type of growth that I think really depicts what would go on in reality, but it does tell us something about population differentiation. A lot of our work we do in raised nursery beds. We find that these beds, we can control moisture in them. Here we've got this PVC rack that we can put rain barriers across. We have barriers to roots at the base of these beds. They're at a great height for working on, not breaking people's backs, doing measurement after measurement after measurement. And this happens to be an experiment of Western larch that is underway, that my PhD candidate in my group, Beth Roskally, is studying for her research. And here you can see Western larch is a deciduous species, of course, unlike most conifers. And here it is just flushing this spring, but they lost all their needles over winter. So from those kinds of experiments, we can then get a good idea of the population differentiation for adaptive traits. And the trait that we see the most differentiation for is cold hardiness. And so this is a map of interior spruce populations. We've got over 250 provenances tested from a common garden experiment. We artificially tested them for cold injury. And the blue dots are very cold hardy. The red dots are very cold sensitive. And you can see a general pattern, geographic pattern here. And that relates to quite closely to climate. And this map shows 30-year extreme minimum temperature, which is a modeled climate variable from climate NA. And so we see the patterns of, makes sense, the provenances that come from very cold places have a better fall cold hardiness than those that come from warmer locations. And we see the same patterns in species after species. Here you see lodgepole pine on the right, interior spruce on the left, and we see very similar patterns. So this suggests that there may be commonalities among species for how we prescribe assisted gene flow, because we've got populations that are dealing with the same climatic conditions and reacting to them in similar ways. Now, when we say that these species are locally adapted, it's a matter of scale. And so, yes, they're locally adapted because they've got huge ranges and we see strong patterns of variation across those ranges. But actually, when we map similarly adapted populations from the data I just showed you for cold hardiness and other growth and phenology traits, we find that these zones of adaptation, if you will, are not small. They're quite large. And so each of these colors represents areas that we find similarly adapted species. These plots out on the side show the average values for 5 different phenotypic traits for each population. And the maps are somewhat similar between species, but certainly not identical. So one of the things we're most interested in with climate change, of course, is drought. And I know in Oregon, you've had some exceptionally dry years in recent years, as we have had in BC. We've had exceptional wildfires. We see drought mortality in tree species that we haven't seen much of previously right here on the coast. And so there's a lot of concern over drought. So we've been doing drought experiments, and this is the work of another PhD candidate in our group, Rafael Candido Ribeiro, and this is one of his drought experiments where he's got trees from about 80 different provenances, and he has them in a greenhouse and he's droughting them. You can see there's control treatments and drought treatments here. And he measured chlorophyll fluorescence throughout this experiment as a measure of the damage to those trees. And interestingly, we don't find the same degree of differentiation, not even close, for drought as we do for cold. Now, this is a map of cold hardiness patterns in Douglas fir. We've got the 2 varieties, coastal Douglas fir and interior Douglas fir, being very different. But we also have variation within each variety for cold hardiness. Whereas when you look at drought, there's a difference between the coast and the interior, with the green, more drought hardy interior Douglas fir and the less drought hardy coastal Douglas fir. But when we look for patterns within variety, we find almost no variation associated with provenance or with climate, the climate of the provenance. We found a similar pattern in lodgepole pine. We found a similar pattern in Douglas fir. We're now testing Western larch, but it is interesting that we do find within population variation for drought, but we don't find strong among population variation. We're also using genomics to identify patterns of local adaptation and also identify genes associated with traits of interest, climate-related traits. I'm not going to go into this much, but we do a lot of genotype environment association testing, where we look for patterns in exome capture data that relate to the environments trees come from. And so typically we do exome-wide capture of up to about 50,000 genes And then we look for patterns in allele frequencies. So one of the things that we found is that genomic data give us quite a similar ranking of climate variables as phenotypic data, and that's quite reassuring. So here we have 19 different climate variables, and this is the amount of climate variation explained in a 20-year-old provenance trial by growth, and this is the amount of climatic variation explained by our genomic data. And what we see is that low temperature variables are strongly associated in both data sets. That is, that you can predict the climate a tree comes from based on its growth phenotype in the provenance trial or based on genomic data. We find that moisture-related variables have weaker signals, but we do find that summer moisture has a signal in the genomic data that we're not picking up in the provenance trial data. And it may relate to longer term, maybe our provenance trials just haven't sampled enough variability in climate. But this is reassuring because certainly we can take genomic approaches and get data quite quickly, as long as we can afford to pay for the data. So Mike Whitlock and I wrote up a paper about the genetic sort of risks and benefits of assisted gene flow, and it all boils down to this graph. I don't want to take too much time to explain this, but I can say that the risks of assisted gene flow are greatest if we've got populations that are long diverged, where we might have outbreeding depression if we combine individuals from very different genetic backgrounds. With our trees, we don't see outbreeding depression. They're wind pollinated, and there's so much gene flow going on that it doesn't seem to be an issue, but that doesn't mean it's not an issue in some systems. So that would be a concern. If we have no genetic differentiation, because we've got lots and lots of gene flow, then there's not much point in assisted gene flow. If you don't have populations that are locally adapted, there's no point in changing where you're getting your seed from. But the important area is where we have sort of intermediate levels of genetic connection between populations. So we don't expect outbreeding depression, but we do expect that we can increase pre-adapted alleles and pre-adapted genotypes. We do have to be careful that we're not disrupting other aspects of local adaptation. So in terms of trees, we've got a pretty good case for assisted gene flow, but you really need to know your organism. And so risks of assisted gene flow, disruption of local adaptation to non-climatic factors like soil type could be an issue for some species. We don't find that signal with widespread conifers, but it doesn't mean that it's not there for some species. Certainly there's uncertainty around climate projections. And certainly if we do this, but there's no need to because there's no population differentiation, then we may well be wasting resources. I throw in this example because this is a very nice study of a grass. And I know some of you work on grasses, but this study showed local adaptation of populations to both local soil type and local mycorrhizal fungi. And so that's a case of where assisted gene flow would disrupt that association. So know your organism. I think that's a really important aspect of all this. So we have strong support for assisted gene flow in British Columbia and across Canada. Some survey work done by colleagues of mine as part of our genomics projects found that in the general public, assisted gene flow was supported as a climate adaptation strategy for forestry by 83% of the respondents in a very large public survey. Assisted species migration, moving species outside of range, only had about 53% support. And of course, I'm sure you're all aware of the ecological uncertainties that are associated with moving species outside of range. This pattern of acceptance was mirrored by forestry professionals and by professional biologists across the country that were also surveyed. Interestingly, there was very high support for local tree breeding, for adaptation to climate, and also for natural regeneration. So reforestation and restoration efforts really need to look at changing seed source. I think this has been quite slow to come in the restoration community. Foresters have been talking about this since maybe the 90s, and it's taken a little while for it to really become reasonably widely accepted as something that should be considered. We need to be careful and not move stuff too far too fast, or it will end in a bad outcome, and it will give the whole endeavor also a bad name. So I think it's really important that we're not targeting, 2050 climates. We're not there yet. Movements outside a range have greater ecological risks and lower support than assisted gene flow. I should say we are moving some species out. The province of BC allows now for the movement of some species outside a range on public lands. We have done an experiment with white bark pine moving outside a range, but that was done to see if it could be done, not to say that we should do it. And we're still tracking that experiment. And mixing provenances may help with risks. And I can talk more about that if people have questions. And I just want to acknowledge the people in my group. We used to be able to crowd together in tight spaces like in this growth chamber for pictures, and now we're reduced to this, but we'll be all back together soon, I hope. And thanks very much for the opportunity to speak today.
Awesome. Thank you, Sally. So we'll go to Tom's talk and then have time for questions afterwards. So just to introduce Tom, so he's the founder and executive director of the Institute for Applied Ecology, a nonprofit organization with a mission to conserve, sorry, I lost my screen. With a mission to conserve native habitats and species through research, restoration, and education. He is also a courtesy associate professor in the Department of Botany and Plant Pathology at Oregon State University. Tom conducts research on rare species reintroductions, habitat restoration, plant invasions, and plant population responses to climate change, and engages prison inmates in conservation through the Sagebrush in Prisons project. Sourcing native plants for restoration is a key area of interest, research, and publication for Dr. Kaye. He serves as a commission member on the IUCN SSC Seed Conservation Specialist Group. And Tom, you could take it away.
Thanks, Gabriella. That was nice. I'm really pleased to be able to be with you all today, and I'm really thrilled as a board member for the Society of Ecological Restoration to be able to talk to a student chapter. So this is a good fit. I'm really pleased. So can you see my slideshow right now? Okay, good. So the title of my talk is Diversity is Magic, Emerging Issues in Selecting Appropriate Native Plants for Ecosystem Restoration. And I want to step back, way back, and point out that we're on this globe, this beautiful blue ball in space that is changing fast. And some folks have put it, we're in the Anthropocene. We've changed the atmosphere so much with greenhouse gases, carbon dioxide, and others that the climate is changing our world, it's changing us. And we need to both try to halt and reverse this trend and cope and adapt to it. A lot depends on that. And it's not just the climate that has changed, but the human footprint. In this paper by Hobbs several years ago, they showed that the human footprint has expanded to such a degree on Earth. And you can see in this top panel, the areas where this index they came up with is really hot. There's a lot of people, and we're changing the landscape and the biota together extremely. And of course, coming along with that are greenhouse gases. And even now, and these are very conservative numbers in my opinion, in the United States, about 5% of our flora is threatened or endangered with extinction. And about 20% of our flora are invasive species. So we're tilting our flora and are the plants that are the base of the food chain in interesting directions here. And that's similar on a global scale. You know, if we look at the IUCN Red List, about 4% of the world plant species are on a very precipitous decline. And again, I think these are conservative numbers. Diversity is really on the run in the world. In this one study from Southern Oregon, where the authors went back to some plots by this old ecologist, Robert Whitaker, in the Siskiyous, they were able to go back to the same places where he put in plots in the 50s and then examine what the plants and things were doing there now. And you know, one of the big messages was that regardless of soil type, there was a huge loss in plant species richness in the plots. So, and the primary change that's been going on here is climate change. There wasn't a lot of other drivers available to explain these differences. So, well, what has this got to do with seeds? My thesis for you today really is that ecosystem service is the goal in restoration, that diversity is magic, and I'll tell you what I mean by that later, and that seeds are the key to unleash this magic and achieve ecosystem services in ecosystems. So really, diversity is the spice of life, I would assert, and the importance of diversity for ecosystem function is something we'll talk about in a minute, but I think it's all important. The importance of native species over weeds or non-native species we'll get into. We'll talk about some tools for increasing diversity. We'll just touch on that really. And then close with some current and emerging issues in seed selection and some online seed selection tools. I'll focus on one of those. So the diversity of hosts improves survival of the threatened golden paintbrush. So this is kind of a one species benefit of having species richness. So this plant called golden paintbrush has an important characteristic and that makes it interact with other organisms in its system, and that is it's a parasite underground. Its roots connect to the roots of other plants. And one thing we found is that as the diversity of neighboring plants within its rooting distance increases, its survival increases. So this is a parasitic plant with a varied diet. And the more species it has to interact with, to draw resources from, the greater its likelihood of surviving from year to year. So this is one example of an ecosystem function of biological diversity, and that's to support an endangered species. But many folks have looked at this from a much larger scale. So this is at Cedar Creek, natural area and research area, where they've looked at the effects of diversity on ecosystem function. And the research there has been largely led by Dave Tillman and others. At least at first, Tillman was really the leader of this research station. And this is where he was able to show the relationship between the richness of species in a plot and ecosystem stability, the variation or the lack of variation from year to year in primary productivity. And I cannot overstate the importance of this finding, that this is at the core of the survival of the Earth, that if we're going to have ecosystems that are stable, that are able to continue with biological productivity over time, those ecosystems need to be biologically diverse. That is the important signal from this study. And then there's another study from a similar kind of setup. This is the Jena experiment in Germany, where they've also looked at the effects of diversity on ecosystem function. And these folks have done some really interesting research and some really great synthesis papers. And this one by Scherber shows the bottom-up effects of plant diversity on kind of all the other organisms in the system. And I want to point out that plant diversity is what they manipulated, and the diversity of everything else is what they measured. And plant diversity is really at the middle of this. There's both below ground and above ground systems that are all affected by plant diversity, not just the identity of the plants, but the diversity of them. And if you look at the top right panel here, just for simplicity, this is what's going on above ground with the diversity, the species richness of the other organisms in the system. And you can see that as plant species richness goes up, the richness of all these other organisms goes up as well, with one exception, and that's the invasive plant species, which so as native plant diversity went up, the invasibility of the system went down. So why native diversity though? Why do native plants matter and non-native plants? I mean, surely invasive species are providing some ecosystem function. And many of them have flowers that support pollinators, etc. Well, let me address it this way. When we look from our own data at the effects of exotic cover on exotic species richness, there's really not much of an effect. But when we look at the effects on the right of exotic cover on native species richness, we see that as the biomass, the cover, the abundance of the invasives goes up, the diversity of the non-natives declines sharply. And so this is not a diversity relationship, it's an abundance diversity relationship. And that's what's driving down native species richness. Okay, well, aren't those natives just being replaced by invasives? Well, no, they're not in this case. And I'm going to move on here. When we look at place-to-place diversity, the so-called beta diversity, we find that the native species have much higher diversity or turnover in species from place to place than the weeds. Another way to put it in plain parlance is, when you go from place to place, there's all this cool diversity in plants, and there's new plants everywhere you go, but it's the same damn weeds every place you go. There's very little turnover. And so this actually has an effect not on the plants, but on those organisms that interact with these communities. And for example, it's been demonstrated with birds, which fly around from place to place and experience the species diversity and the species turnover, that species diversity of birds is negatively impacted when the beta diversity is low as well, and when that's driven by weeds. So yes, weeds can provide some ecosystem functions, but not to the degree that native species do. And it's that diversity of native species at a site and turnover from sites that adds that magic. So how do we restore diversity? Well, really with seeds. It comes down to seeds. And seeding in some ways may be the only way to increase diversity in some systems. Without seeding, we can't increase the number of species that occur at certain sites. Now, yes, sometimes there's a species, a seed bank present, so when you disturb it, some natives come out of the ground. But locally here in the Willamette Valley and the Puget Sound prairies, that is not the case. We have very weak native plant seed banks. And well, we can plant plugs too. We can get plugs to go out there, but you can't do that at a huge scale. So seeds are crucial, and we need to go to a seed-based restorations in order to do this at any kind of scale and to restore diversity. So adding seeds does add diversity. We've done a number of experiments on this, and I'll just cover one very briefly here. This was an experiment we did about 15 years ago at 10 common research sites throughout Oregon, Washington, and British Columbia. And this involved a number of different kinds of treatments, but I'm just going to pull out a piece of that here to demonstrate that the year after a burn, or the year of a burn, the season after the burn, if we seed then, we get a big boost in native species richness. And without that burning, we don't get quite as much of a boost. And when we do that seeding at a different time, a year after the burn, we don't get nearly the boost either. So how we do restoration matters, and timing of seeding in regards to when treatments like burning or herbicide or whatever has a huge effect on the success of those plants as coming up from seed. Of course, that has to do with seed soil contact and various agronomic issues. But it's important that we pay attention to our techniques and put our seeds out when they're most likely to result in better seed production or better establishment. And one thing I wanted to point out was in the same study, we had some collaborators come back several years later to the same plots and look for evidence that the treatments had lasting effects. And the only treatment that had a lasting effect that was really strongly detectable years later was seed addition, not whether it was burned or sprayed or something like that. Because of the scale the project was done at, those treatment effects kind of faded with time. But seeding had a lasting effect. And so that's one of the take homes, is that of all the things we do, adding seeds is the kind of thing that increases diversity, not necessarily the on-site management without seeding. So this is a truism practically. Plants vary, right? So when we pick plants to put out a site and we pick the genetics, we need to be considering those genetics because they're going to interact with the site and its restoration and its climate. Local adaptation is the general rule in plants. In this recent review of local adaptation, one of the things we found was that there was significant measurable variation among populations in 305 common garden studies. So variation among populations is the rule, it's not the exception. 95% of common gardens found this. And of course, you know, that may seem just, of course, obviously things vary, but this is the stuff of natural selection. This is the grist for evolution and for how we can leverage that information and put it into restoration. And 86% of the studies that looked at this had trait by environment variation. So this variation is not random. It is connected to natural selection around the environment. And 75% of those environmental variables that were climate related. So climate is the big driver in local adaptation. And in those few studies where we could look for local advantage, we found that the local populations in a common garden had higher survival, 67% of the time, and better reproduction, 90% of the time. So local adaptation is there, it's driven by climate, and it results in better survival and flowering if you use the locally adapted type. That all makes sense. And so how do we manage that then in a restoration context? Well, one approach is to use seed transfer zones. So this is the idea that you can draw a circle on a map or a boundary on a map and say, everything within this area has a similar enough environment that if you move the seeds around of a single species, that they should be relatively equally adapted. Okay, that's the concept. So you don't have to worry about where it came from as long as you're within that polygon. That's the pitch anyway. These provisional seed zones for the United States are released in an effort to make this kind of information available to practitioners in the absence of better data. And for the vast majority of species, there's no data at all. So this is important and useful. But we also must understand that this is provisional, and for some species where we have better data, we should use that. That, you know, don't dumb it down when you have the better information. But this is an approach that we can take. And then, you know, planning for climate change is a piece of this too. So we know the landscape is changing out from under our feet or our roots or our leaves as we go forward here. And that means that the optimal environments may shift over the landscape. So how do we what are some strategies for obtaining seeds? Well, we can keep them local, strictly local, like we're going to only restore with seeds that were collected on that site. Okay, well, that's pretty strict, and there are a number of people that operate that way. And We can have more relaxed or mixed local, where it's sort of from within the local area, but doesn't have to be on site. We can have a composite mix where we've got a few different populations. We can have a range-wide admixture, which is you go throughout the distribution of a species and you bring in genes from all over the place. Or you can have a predictive, a matching to future climate approach. And I'll keep moving here. So in practice, what most people seem to be advocating at this time are either restore with high genetic diversity, some kind of pooled genetics within your area, or import your genes from a place where your climate is now producing seeds. So, you do a future forecast and see where on the landscape is my future climate, or perhaps even my current climate, and get genes from that area and bring those in. And of course, these are not mutually exclusive. You can have a combination of the two. And that's personally what I advocate for at this time. Now, at the Institute for Applied Ecology, we help facilitate some seed partnerships in the Willamette Valley. And one of the approaches we take to collecting seeds and putting them into production is to collect seeds from lots of different places within the Willamette Valley, grow those out together in an agricultural environment, and then harvest those seeds. And then, one of the questions might be, does that result in higher genetic diversity or improved adaptation? Does it lower inbreeding? Does it reduce pressure on any one wild population? And the answer to those questions is yes. And we've been able to do some research in an example with golden paintbrush again, where we were able to look at the genetic diversity in wild populations, then collect seeds from those wild populations, put them into a nursery and track them there as well, and then take the harvest from the nursery and put it on the ground and see, did we actually increase diversity as we thought, or did we have something happen in the nursery that might have resulted in domestication selection or narrowing of adaptive traits? And that was not the case. So in general, we can achieve that. The downside could be outbreeding depression, but as Sally said, it's not very likely for most species. It's only in those extreme situations. And most of the practitioners I know are not advocating for any kind of extreme long distance movement of seeds. So I don't want to dismiss outbreeding depression, but I think it can be overstated as a concern. I'm going to skip this slide and point out that when we get into restoration in a changing climate where some species simply can't grow in an area anymore, what do we do? The mixture, the community assembly is perhaps no longer what we could have achieved if we were looking in the past. So we can't use historic community types necessarily as what we want to plant now, because the rules have changed. One approach I'm advocating is use native species and emphasize local species in your designing your restoration, but use those species that are now the most common and the most frequent and most abundant, because they're obviously the winners already, right? They have weathered the climate change and the invasion by new competitors. So they're not naive to this environment, but we may put them together in a new package. We may allow them to, or ask them to coexist in a new manner, but they're already the winners. Those are our workhorses that we can consider using. So some online tools. The one I want to talk about is the Climate Smart Seedlot selection tool. And so I'm not involved in the production of this. This isn't a commercial, but I think it's a very useful concept. And they've put together a tool that's online that anybody can use. And I think that is a good place for us to start and have this conversation. But the idea is you can say, here I am on the landscape. Show me where the climate I will experience in, say, mid-century is today. So I can go get genes or seeds or cuttings or whatever from that area and start incorporating them into my restoration today. So these are my future climate analogs. And so how can you find those? So this is a tool that can allow you to do that. They use 15 GCMs that are combined and averaged, et cetera. So it is what it is. And we're working on a project with some folks in Northern Oregon around Portland that want to do riparian restoration, but they want to have a climate plan in place as well. And so we've used this tool to forecast where on the landscape we might collect material that represents what the climate in this Portland area will look like mid-century versus late century. And what's interesting about this is it's essentially the same picture as you look forward in time, but it's more contracted to this Northern California area. And it's not always how that works, but that's what's going on in this rendition. And we're asking now, there are contractors out in the field gathering data from these areas that we've identified might be future analogs to look at what are the species that occur in these areas, what do those communities look like, so that we can not only be asking what genes can we be bringing into an area, but what new species might we want to consider bringing into this area. Now, this is pretty bold, perhaps stepping pretty far for now, but it's an exercise to think ahead. And in some of our conversations, we've talked about, well, you look down in Northern California where it's really the best match. There's a lot of redwoods down there. Let's plant redwoods up by Portland. And it comes out as like, well, yeah, wait, are you serious? And the answer is, I don't know. But if we're thinking about moving whole species, something like a redwood, which is pretty big and bold, might need to be on the table. We need to have the conversation at least and find out where our boundaries are as a community of restorationists and the public and explore this. But mainly, we want to be thinking about moving genes. And I don't mean replacing what we're planting with Californian genotypes. I mean a judicious admixture, a dash of this and a dash of that, so that we're strengthening the local genotype with diversity that contains adaptation to the future. And I think that seems like a cautious minimum amount of effort that we can put into this. So I'll close with that. I have no idea what the time was, but I'll stop here. Thank you.
Thanks to both of our speakers. Those are great presentations. We are at 5:22. If you both could stick around, we do have a few great questions in the chat, and we might have some more in the audience too. If anybody has to take off early, we are recording this conversation, and we will have the whole thing up on our website, hopefully within a couple days. So if that sounds good, maybe I can start with the questions for Dr. Aitken, and Gabby, then you can go over the questions for Dr. Kaye. So I think I have two questions here that I can, sorry, I just have to hide the participants, that I can combine related to cold hardiness. The first is generally why cold hardiness is important to consider for seed sourcing is if climate is generally warming. Mm-hmm. And then how that relates to extremes. So how do we balance that idea that there's overall warming, but potential for extreme cold snaps in terms of sourcing? Yeah,
so that's a really good question. First of all, we get the strongest signal of local adaptation from cold hardiness, and it turns out that there's a pretty tight linkage between the set of genes that we find are most strongly associated with cold hardiness and also growth, bud phenology, so timing of bud break, timing of bud set. So this whole sort of adaptive complex, but is tied in most strongly to cold. And if you think about a little seedling that you plant or that naturally regenerates is going to be at its most sensitive stage to cold. So a big tree can take more cold than a little tree during active growth or during the regular growing season. And so if we're moving trees very far, the climates we move them to are going to be coldest the first decade we move the trees there, and they're also going to be the most cold sensitive or susceptible that first decade. So it's something we just keep it gives us a good indicator of the strength of local adaptation. And it's kind of a warning flag that we shouldn't move things too far, too fast. I think that that's the struggle we're in, because we've got this moving target. But if we go too far, we're going to end up having cold injury. And so that is one reason. Another is this point of extremes. And so when we see this strong pattern of local adaptation for cold, is that the result of just regular kind of every year cold, or are we seeing the effects of episodic selection for really extreme events? And we do know that climate change is coming with a lot of variability in climate. So we want to make sure the trees can survive. So it is counterintuitive. Why do you care about cold if the climate's warming? But we just want to be judicious, and that's why this idea of mixing genotypes might be a good solution. And I didn't talk about species diversity, but species diversity is the first thing we should be doing with restoration and with reforestation and everything else. So I really enjoyed Tom's talk along those lines.
We have another question. Are there any hypotheses for why there is a weaker provenance variation for drought tolerance?
Yeah, I have a few hypotheses for that. And I'm glad you brought it up. First of all, if you think about water availability, like the populations, it's not the amount of water that's falling from the sky that matters, it's how much water is available to them. And the spatial variability in water in the soil, available water in the soil, we have a super mountainous province, and as you've got a lot of mountains down there. And so water availability really depends on slope position, soil depth, soil texture, all these things. And it may be that is too variable on too fine a scale for populations to adapt to, because you have to think that gene flow, pollen's going everywhere. So if we've got a really small scale of environmental variability, you get adaptation to the general environment, not to those specialized environments. That's one thing. That's one possible explanation. The other is that there's just really good global drought hardiness in these species. And so the whole species is drought hardy enough that there hasn't needed to be that local adaptation. The third possibility is that, to put it bluntly, that maybe we suck at phenotyping for cold. And that's something we talk about all the time in our lab. And I'm really happy to have Beth Roskelley working on this. She's done some cool work when she was in Montana on actually mechanisms in the wood, in xylem, that seem to be associated with drought tolerance. And she's going to be torturing and killing large trees all summer and measuring them. So great questions.
Cool, and we have, I think, 2 questions for Tom. So one is about, someone thought the diversity supporting the paintbrush was really cool. I was wondering if you have thoughts about the mechanism there.
Well, yeah, I have thoughts, and I think it has to do with the fact that this is a parasitic plant that is drawing resources from multiple species. And, first off, one thing to keep in mind is the graph I showed you where there was the relationship between diversity and survival was for perennial hosts, and that was a positive relationship. If we look at the diversity of annuals in the same plots, it's a negative relationship. So, and well, why is that? Because probably if you're a perennial parasite and you connect your roots to an annual plant that dies, you die too. So you invested resources in that connection, and you threw good money after that, so to speak. And then when it comes to what is the diversity of perennials doing for you, I think this is the fact that not all plants are equally good hosts. So as you increase the number of hosts you have the likelihood that you'll have a really good host in that mix will go up. So that's the sampling effect explanation for why diversity increases ecosystem function. But there's also the complementarity hypothesis, and these are not mutually exclusive. And the idea is that as you have more hosts, those hosts may be doing different things for you. So, or at different times of the year, because they may be active at different times of the year. So from one host, you may be really good at drawing moisture, and from another, you may be better at getting nutrients. And from a third, you might be getting secondary compounds that are conferring a defense against herbivores, which is something that castellators can do from hosts like lupins. So, just having a lot of different hosts means you get to, draw from different currencies, and you improve your portfolio of resources. So I think it's a mix, potentially, of these different mechanisms.
And then the other question is, why are seed zones so much bigger in the east? And does it have to do with data availability, as you were saying, or something else?
Yeah, it has more to do with the lack of mountainous and topographic variation. I don't think it's a lack of data. I think it's topography and soil type variation. There's just much more continental wrinkling and rumpling in the west than there is in the east, and that results in a more complicated soil and climate interaction. And so I think it's plain and simple, has to do with how bumpy the ground is.
Awesome. And I think the last question seems to be for both of y'all, maybe. What pushback have you encountered when introducing non-native species that would increase diversity and survivability to new environments, and how do you respond?
Haven't done it yet, so I'm not really sure how people will react, but I brought up that topic of maybe planting redwoods intentionally in riparian areas near Portland. I mean, you can imagine that some people will go, that's cool, and others will go, over my dead body. So it's going to be an interesting experiment in sociology as well as ecology.
There's an island near here, just east of this island, called Cortez Island, where there's a large property being planted largely to redwoods, also giant sequoias, dawn redwoods. And it's sort of this rewilding thing. I haven't visited, but I'm curious. Anyways, the only thing that we've moved out of range is white bark pine for this experiment we did that was seed based. So we planted 18,000 seeds in 9 sites. Up 6 of those were out of the species range, up to the border with Yukon. Now, any of you know white bark pine? After about 5 years, the trees were about this big. So the idea of invasion, it would take it would take many centuries. And I think you'd have to ask if it was a success or a failure if you had something you called an invasion with something that grows so slowly. White bark pine's listed as endangered in Canada under our Species at Risk Act. And we, as I mentioned before, we did that to see if it could be done, not to actually move it. And we didn't get much. We were very careful in the paper to not advocate for actually doing this and saying that there really needed to be a more, thorough assessment. There was an ethical paper written on it asking if it was an ethical decision. And they came to the conclusion that it would be ethical to move that particular species, given the risks and the benefits. But I think that I'm much more on the side of when people get gung-ho and start talking about moving things outside of range, I'm pretty cautious about actually implementing that. And I'm cautious about it under our provincial policies. I've been sort of an advocate for slowing that process down. So, yeah, but I think what it comes down to with white bark pine is in terms of the feedback that we got, including from an Indigenous group up in the areas we were moving, that we had an experiment in, as well as information from Indigenous groups within the native range of white bark pine, was that if what you really love is the ecosystem as is in an area, then you are really going to question moving that species in. But if what you really love is whitebark pine, then you're probably going to think that trying out assisted migration as a conservation tool is going to be a good idea. And so it comes down to people's values and perceptions and experiences. And those things are much trickier with things moved outside of range, as the social scientists that I showed one graph from have shown. But it's a really interesting area. And if you want to read more about those social aspects, human dimensions, check out Shannon Hagerman check out her Google Scholar page. She writes a lot about climate change and conservation and different reactions to different approaches. And she's just come up with a new framework around resistance, resilience, and transformation in conservation practice. So she's doing really great work in that area. She's a collaborator. What was that, Tom? Again? Shannon Hagerman. I'll put it in the chat. Great.
We did a survey about 10 years ago about restoration and climate change. And one of the questions we asked was sort of a pair of how open are you to moving to a new area to save it, a species that occurs in your area, versus receiving a species from some other area. And there was a small but real difference in that more people were interested in saving their species than they were in receiving somebody else's and taking the risks that might come with that. So it's an interesting, there's all this perception and emotion part of this that's very real. And it's hard to separate out from the conservation values and what are you trying to achieve in the end? And that's what we need to focus on perhaps more, so that we can step back from our emotions and think about how we're going to get someplace that's going to be good for the planet. And that's not easy. And we don't even know the answer. So, we just have to culturally and socially muddle through this, I think.
One person who's done some interesting writing on this topic is Emma Morris, and she's in Oregon. She wrote a book called Rambunctious Garden, I think.
Yeah.
And she's pretty good at taking a hard look at how we perceive ecosystems as how we perceive how ecosystems were before European colonists arrived, and sort of that whole history. And she really advocates for diversity in whatever form it comes from. So, she takes arguments to a point where I wouldn't, but I think that she goes through a lot of really interesting thoughts along the way. And she's a very good writer. She lives somewhere in Southern Oregon.
That's another really good reference.
I just have one more short question from Sarah, and then I think if anybody else has any other questions, maybe we can collect them and send them to our speakers, get answers by email. This is just a short question following up on the paintbrush question from before. Sarah's wondering if the paintbrush is benefiting from species richness, or it's more from functional diversity.
But in the paintbrush example, I think it's likely functional diversity. But as the species richness goes up, the functional diversity also goes up. And part of the question was, are those mostly grasses that are the perennials? And the answer is no. There's a mix of grasses and forbs. So it's quite diverse. And another kind of analysis we did was to look at, are there certain indicators of survival within a site, and how do those vary from site to site? And the bottom line is, those species of potential hosts that are indicators of survival at one site are not the ones at the next site. there's species turnover and change and replacement in function, perhaps. So there's a lot of variation in how golden paintbrush is experiencing its neighbors from place to place.
Thanks. Well, thanks to both of you, Dr. Kaye, Dr. Aitken, for having this talk with us today and sticking around a little bit longer than expected. If we get any other questions, we will be sure to reach out to you, and we'll have this discussion recorded on our website. And thanks to everybody for joining us today.